today's episode of Myeloma Crowd Radio, a show that connects patients with myeloma researchers. I'm your host, Jenny Ahlstrom, and we'd like to thank our episode sponsor, Celgene Corporation, for their support of Myeloma Crowd Radio. Now, today is the second show in a series of 3 on Myeloma Crowd Radio for the Myeloma Crowd Research Initiative, or as we call it, the MCRI. Now, when we started the Crowd Care Foundation and the Myeloma Crowd, we asked our scientific advisory board what we could do to fill gaps in research funding. And the first time that we did this, they told us we should fund high-risk research. So we ran the very first ever crowdsourcing and crowdfunding campaign, and we picked 2 immunotherapy projects. The first was a CAR T project from the University of Würzburg in Germany that was targeting CAR T cells with CS1 and BCMA, and another immunotherapy called MILS from Dr. Yvonne Borello, an immunologist at Johns Hopkins. The second year we asked this question, Dr. Fonseca suggested that with all the new treatments, we should create a tool to help patients identify treatment options they could consider, especially if they were being seen in the local oncology setting. So based on this need, we developed the HealthTree tool. And with HealthTree, patients can, number 1, understand their personalized treatment options to have better and more informed discussions with their doctors. Second, they can find clinical trials they're eligible to join based on their lab values and all the eligibility criteria. And third, they can share their myeloma experience anonymously with other patients and researchers to help identify faster cures. So in the summer, we canvassed 50 cities to share this tool with over 100 patients. And now over 2,200 patients are using HealthTree. And so we're very excited. We've just begun showing some of the reporting in that with who is using HealthTree, which is wonderful. And today we'd like to announce a new feature in HealthTree that lets you write your personal myeloma story so you can share that later with your doctor or with other patients if you so choose. As we think about this, there's a lot of focus on precision medicine, but personalized medicine includes not only your myeloma genetics and your labs and your personal experience with the disease, like your family situation or how your feelings about how you're dealing with this disease. Now, our focus for the second Myeloma Crowd Research Initiative is complementary to what we built with HealthTree. We heard from doctors that a small fraction of patients could be cured, and we want to know who they are and how they're achieving success so we can replicate it for other patients. So the second MCRI is focusing on optimizing myeloma care because so much can be done to lengthen life today with today's drugs if they're applied for the right patient in the right order at the right time. And we've asked all 3 projects to integrate in some way with HealthTree. Now, last week we heard from David Zhan from Memorial Sloan Kettering on his testing of the immune system to create an immune system signature. Today's show is about identifying personal strategies to prevent relapse or to kill all the myeloma from the beginning. And on Friday, we'll be sharing our 3rd project of 3D tumor modeling. So each of these 3 projects bring a personalized approach to caring for you as a myeloma patient. We'll be funding these projects for a total of $500,000 or more and invite you to join us. You can go to give.crowdcare.org/MCRI and create an individual or team fundraising page. Then share it in any way you want. Join a local run or walk and share your page. Share one of these interviews on social media during the holidays as a way people can support you in your efforts to speed up a cure for myeloma. You can share by email, Twitter, Facebook, or other social media accounts. And we know that your friends and family want to know how to support you during your disease journey, and this is an excellent way of doing that. And the end of the year is a great time to do that as well. So now we would like to welcome 2 investigators to the program today, both at the University of Iowa, who are working in tandem to make this research a reality, Dr. Frank Zhan and Dr. Yogesh. Jethava, welcome to the program.
Hi. Good morning. This is Frank Zhan. I'm very excited to discuss my project with all of you.
Good morning. This is Yogesh Jethava, and I'm as well excited to discuss the project and the future prospects.
Well, let me introduce you both before we get started. Dr. Frank Zhan is Professor of Medicine at the University of Iowa in the Hematology Bone Marrow Transplant Department since 2012. He's also Adjunct Professor of the College of Dentistry and Dental Clinics since 2016. Previously, he performed research at the University of Utah for myeloma at Huntsman Cancer Institute, as well as the University of Arkansas Medical Sciences, or UAMS. He helps review grants to help those identify those that should be approved for several foundations, including the National Cancer Institute for R01 and R21 grants. These are the largest grants and the most prestigious grants from the NCI. He's also on the editorial board of journals like International Blood Research and Reviews, the American Journal of Blood Research, and the Journal of Blood and Lymph, and others. His current grants that he's received include this one, and the role of NEK2 in myeloma residual disease, genetic studies of myeloma genes, high-risk myeloma approaches, and the use of vitamin C infusion in myeloma, and what we'll be talking about today, the identification of myeloma stem cells. Dr. Zhan has very deep research expertise on myeloma genetics, having over 2 decades of study in that field. Dr. Yogesh Jethava is Associate Professor and Director of the Multiple Myeloma Program, as well as Associate Director of the Blood and Marrow Transplant Program at the University of Iowa. Prior to this appointment, he was Director of the Bone Marrow and Stem Cell Transplant Services Division of Hematology Oncology at the Winthrop P. Rockefeller Cancer Institute at University of Arkansas for Medical Sciences, or UAMS, where he established the first fully functional allo transplant unit at this, and he was director and attending physician for the Leukemia and Lymphoma subspecialty clinic there. In his work, Dr. Jethava is an International Myeloma Working Group member, FACT inspector. He's part of the EBMT Malignancy Working Group and a member of the Experimental Therapeutics Groups and others. He reviews abstracts for publications like Blood Leukemia and the Blood Cancer Journal, and he's also working on studies that involve high-dose vitamin C infusions for plasma cell disorders like myeloma, and expects a trial to open soon at the University of Iowa. So thank you to both of you for joining us. We're thrilled to be able to fund some of your research on these important topics. So let's get started with some of the basics, like maybe the most basic question is, what are myeloma stem cells?
Okay, let me explain this. We think myeloma stem cells. Hello,
Dr. Zhan, if you could go closer, a little bit closer, that would be great.
For sure. Okay. Can you hear me now?
Yep.
Okay. Yeah. Myeloma stem cells is similar to other cancer stem cells. Sometimes also called tumor-initiating cells. That's originally documented and described in leukemia as a real population of cells with unlimited self-renewal capabilities. Then recently, tumor-initiating cells or myeloma stem cells have identified in a number of other cancer, actually tumor-initiating cells in other hematological and solid tumor. And these cells have the biggest feature is the drug resistance or chemotherapy resistance. And the treatment failure is in this cancer, including multiple myeloma, is most likely due to the persistent or minor population of cancer stem cells, which is non-cycling or low cycling and very drug-resistant myeloma population. Okay?
So stem cells are these early precursor cells that become other types of cells in the future, right?
Right,
and
right.
as they mature, because ultimately they become some of them become plasma cells, right, that we're familiar with in myeloma.
Right. So that is a very interesting fact you pointed out, Jenny. But that is a different type of stem cell that you are mentioning. It's a hemopoietic stem cell. Okay.
Oh, okay.
So the way our blood, like say in our blood there are red cells, white cells, platelets, and all other components, they come from hemopoietic stem cell, which is the originating cell. Now there is increasing belief that cancer also arises from one particular cell which has capacity to keep growing and be drug resistant, and that is called cancer stem cell.
Oh, okay. I was not familiar with that.
Yep. So what you mentioned, that's the hemopoietic stem cell. That's a completely different thing. What cancer stem cells? So stem cell basically is the feature. It's the feature of renewability, continuous renewability, and we now believe that lots of hematological cancers and solid tumors arise from a cell which is an originating cell, like a tumor initiating cell or cancer stem cell, as Dr. Zhan mentioned.
Mhm. And Dr. Zhan, you mentioned that they were resistant to chemotherapy, but can you go into a little bit more about, or both of you, about why why they're chemo resistant? What makes them different from these other types of stem cells?
Right. So firstly, this sort of myeloma stem cells have a special expression network. As you mentioned earlier, that induced pluripotent stem cells or embryonic stem cells. Those cells have special expression genes such as ACT4, SOX2 analog, etc. And those sort of myeloma stem cells also express these genes. And these genes can drive myeloma cells self-renew. That's one major reason. Secondly, there are some drug pump protein are highly expressed in myeloma stem cells. So those pump protein can pump out the chemotherapy drug outside the cancer cells. In other words, this drug cannot stay in tumor cells for a long time, just a very short time. That's why they cannot play effective way to kill tumor cells. I think that's the major reason.
Mm-hmm. Because the drug can't stay in the tumor cell, is what you're saying? So
right.
It's not as likely to be able to kill it?
Correct. As Dr. Zhan mentioned, see, there are certain reasons why we call cells as stem cell. So the first and important thing is that they have higher expression of genes, which Dr. Zhan mentioned, and these genes are responsible for self-renewal. So they keep replicating, and it means that the disease persists. And second reason is that they also have some protein on their surface, which basically pumps out the chemotherapy drug out of the cells. So that way, the cells become resistant to the chemotherapy. So it's like a double whammy. There is this pump which is pushing out all the chemotherapy from the cell into outside the cell, so the cells are not effectively killed. Plus, they have this higher expression of genes, which will keep them replicating. So they are chemo-resistant and they keep replicating. And that is the reason why these cells persist for longer duration. Right.
Okay, sorry, I'm taking notes. I think,
no problem.
that makes a lot of sense, that they have this double hit, basically. And that's why they're so resistant to chemotherapy cells. When you look at, does everybody have these cancer stem cells in them, and then some people, their immune system is just able to kill it, or is this just something unique to myeloma where these cells just start, they generate somehow, and then, they keep proliferating, like you were saying, and then they don't respond to the, in the general population, do these types of stem cells exist, I guess is my question.
It's still debating about myeloma stem cells. Some people believe there's more population like cancer stem cells. They can self-renew and drug resistance. Some physicians or scientists, they don't believe myeloma stem cell exists in this disease. However, based on our almost 20 years experience, especially for using gene expression profiling, we found some patients, even in the longer remission, such total CRP1 patients treated at Little Rock UMS, So those patients can survive over 10 years or 15 years. We collected those samples and did a microarray gene expression profiling. We found those translocation genetic change exist on those of complete remission samples. So those suggest us at least a small population exists even in the longer complete remission samples. So from that way, then I start our project with Dr. Guido Trico when we moved to the University of Utah almost 10 years ago. We started to identify or define drug-resistant cells or signaling pathways or sort of myeloma stem cells. So since then, we spent at least 5 to 6 years identified the current project, CD24, has the feature related to drug resistance, cell-free renewal, and the sort of myeloma stem cell feature. Come back to your question. I believe most of cancer, I think, have the universal cancer stem cells. And probably that's the major reason for the disease recurrence or relapse. You may have, Dr. Yogesh, you may have.
Yeah. So as so coming to your question, Jenny, your question was that every cancer has stem cell, right? That was your question. So so there are two philosophies here, and and the jury is out there regarding this thing. But there is increasing evidence to suggest that cancer arises from tumor-initiating cell. We can call it as tumor-initiating cell or cancer stem cell. So is there a 100% evidence? Probably no, not right now. However, increasingly people in the field believe that that is the reason why cancer occurs, that there is a tumor-initiating cell which causes cancer, but that cell is very difficult to get rid of, and that is what leads to persistent disease and eventual relapse of the disease. So, so this whole research started by Dr. Zhan and Dr. Guido Trico from the original data that was generated on total therapy trials. I don't know whether you are familiar with that terminology or not, but just to briefly explain on what total therapy trial is. So total therapy means using all drugs upfront for treatment of myeloma, all available drugs upfront. And this was started by Dr. Bart Barloghi and Dr. Guido Trico in Arkansas. And all patients were treated in similar fashion. They had gene expression profiling tested at regular intervals. And what Dr. Zhan and Dr. Trico found was there was certain number of patients who were in complete remission for say 10-15 years. They had this particular phenotype of cells in their bone marrow, and that phenotype persisted for long, and that led to subsequent research in last 10 years, which is now what we are seeing, this concept of myeloma tumor initiating cell. So that's the summary of the whole thing.
And I think the benefit of using the data from the University of Arkansas was that you had this really long-term data. When you're saying over 10 or 15 years that you're following people and you're looking at patients in remission, and then you're able to test their genetics before and after, and that's pretty amazing.
Right.
That you have that long of data to
very unique,
see.
exactly. So we have patients who are going on for 15 years and 20 years, and we have their genetic data, and that's what enables us to do this kind of research. Yeah.
So when you said you were looking at these patients over 10 to 15 years, it sounded to me like you were saying that you were able to see their genetic changes, and that patients who were lasting that long had similar genetic features from when they started. Is that what you were saying, or were you saying something else?
So for this particular question regarding the total CYP1 patients, actually we don't have a sample collected at the diagnosis because when we start our gene expression profiling, those patients are already treated. So this I mentioned that patient survival over 10 years. That's only have after you get a sample in remission. However, we collect sample in total CB2. Then we have a sample at the diagnostic stage in remission and it relapse. We confirm our observation for these genetic changes. Yeah.
Yeah, that makes sense. So for the first group, you didn't have a starting genetic profile because maybe you didn't have the test out yet, but then the second group did. You know, they had the before and after, which I think is really important. And you've done so much work on the genetics. It's been
Thank you.
Kind of some of your focus, and a lot of your focus, and you've done a great work there.
Thank you.
So we are thankful to you both. So let's talk about relapse after therapy. So if you have these tumor-initiating cells or myeloma-initiating cells that causes the myeloma or the cancer at the beginning, and then you have, but these cells are both resistant and then keep replicating. So you give the patient, you know, let's say you do induction therapy and a stem cell transplant, you do maintenance therapy, and then they still relapse. How do you find these? I mean, we'll talk about this in a minute, but how do you determine, like, who might be more prone to do that, to relapse, or if these cells really are in each patient, and how do you classify them? And anyway, is there any way of understanding who is likely to relapse when you look at things like this?
Right. So yes, so we recently, once we identified this CD24 as a cell surface marker related to drug resistance or stem cell feature, we checked this marker expression in newly diagnosed patients and also patients after treatment. So from current our data, of course, the number is not big yet. I think we had totally 60 samples, including 28 from newly diagnosed patients, 32 from patients after treatment. So we observed overall CD24 positive population, the percentage is relatively higher after treatment compared to sample at baseline or at a newly diagnostic stage. So this sort of fit our hypothesis because we saw that this CD24 positive myeloma cells are drug resistant. So after treatment, these cells can survive and enriched, and that's why these cells can be detected relatively high percentage after treatment or even after stem cell transplant. Then this population can stay for a long time or relatively short time. We don't know for remission. Depend on multiple factors. That you have the intrinsic, like the genetic change factors. Also, you have the microenvironment, like a bone marrow niche or immune response, make this population to grow faster, then get a disease relapse. So we try to say is if anyway we can depend on this phenotypic CD24 population can predict a patient stay longer or shorter in remission, then faster or slow for relapse. That's one goal in this proposal too.
Well, yeah. When we talk about it to patients, I think some patients might not understand what some of these CD things are. So can you explain maybe how you pick this up in the flow cytometry test and what, because most patients will go and get their flow cytometry test run, and it'll come back with things like, oh, you have presence of CD56 or you have presence of CD138 or things like that. And I think they're probably more familiar with some of those, even though most of us have no idea what they mean for our myeloma.
That's right.
But this is one of those other markers, right, that you can find on the surface of myeloma cells?
That's right.
Correct. So
Mhm.
what CD is basically, every cell in our body has a unique CD marker on the top of it, with which, because of which we are able to recognize the cell. So myeloma cell has certain markers on the surface which tells us that they are myeloma cells, and
Mhm.
one of them is CD38, which you mentioned, CD138, CD19. These are all myeloma cell markers. Now, on the surface of these myeloma cells, there is this another marker also, which is CD24, and this is what we are interested in. So, and this test is called flow cytometry, which is a part of bone marrow examination. So, whenever a patient gets bone marrow done, there are certain things we do. One is we see the bone marrow under the microscope and see what is the percentage of cancer cells. Second thing we do is do a flow cytometry, and that means we run this bone marrow sample through a machine, which will pick up these CD markers and will tell us that, okay, this patient has, say, CD38, CD138 positivity, which is basically tells us more specific details about the cancer cells. And then lastly, we also do something called FISH testing, which is basically to see the chromosomal rearrangement on the cancer cells, in the cancer cells. So these are the three standard tests done for any bone marrow. So CD marker is usually picked up by flow cytometry, and CD24 is one of these markers which we are interested in. And
Mhm.
as Dr. Zhan mentioned that what our initial data um, shows, I mean, this is what we are planning to do, like see the CD marker in the beginning of the treatment, after the treatment, how many cells, CD24 positive cells persist, what is the correlation with the relapse. So, so that's our hypothesis.
Mhm. And let me ask you this, because I think sometimes you look at genetics and they affect certain proteins on the surface of myeloma cells. So patients have heard a lot about things like BCMA and CS1 because elotuzumab is going after that. These CD markers aren't necessarily protein targets, or how are they? How are they? They're just what you kind of just explained, right? They're just markers, but they're not. But they are targets
Right.
too, right? Because
Right.
uh-huh.
Yeah. So this is yes, the cell surface marker is relatively easy to be targeted because scientists can develop antibody or CAR T to target the cell surface marker easily than intracellular genes or proteins.
Right.
That's why the current CAR T, like CAR T19 or BCMA, that's on cell surface marker. And the same, that's our goal. If we prove CD24 is a myeloma stem cell marker, the next step we try to do, one is develop a specific antibody to target this cell surface marker CD24 block its signaling pathway, or combine to design for CAR T CD24 or bispecific antibody specifically recognize myeloma cells and also CD24. So that's our goal. We try to do it on this way.
Mhm. So in proteins, when you talk about myeloma genetics and you say, like, let's say gene deletion 17 affects this TP53 gene, and that gene does something really specific, like it's the master cell regulator of death, for all the other cells, which is why that genetic feature is usually aggressive in myeloma. What does CD24 do? Like, does it have a specific function? I'm just curious about that.
So CD24 is not a gene. It is
Right,
basically
or a yeah.
on the presence of, it is present on the cell surface. So what it
Okay.
does is that it gives this overexpression, or say more population of CD24 cell. It leads to more cell survival. So what happens is that CD24 peptide, which is basically present on the cell surface, it activates multiple signaling pathways within the cancer, which is within the plasma cells, and these pathways are called as MAPK, NF kappa beta, Notch, hedgehog signaling pathway. So this CD24 peptide is associated with activation of all these multiple downstream pathways, which leads to active cancer cells, which leads to basically the resistance in the cancer stem cells.
Yeah, regarding your question about the connection with correlation with clinical or genetic change, actually, as I mentioned earlier, we only did 28 newly diagnosed patients, so we don't have data to test the correlation between genetic change with CD24 expression in this moment. Yeah, another point, I think CD24, if our hypothesis is correct, this only expressed on a small population of myeloma cells from the same patient. So that means even you have the translocation or without the translocation or deletion P53, maybe they are difficult to tell is any association between this genetic change. However, recently we did some interesting study when we co-culture myeloma cells with stroma cells. We can see cell surface CD24 expression relatively increased. And also, as I mentioned earlier, the drug treatment also can enrich CD24 positive myeloma cells. Of course, this probably killed that CD24 negative cells. So that's on the change is sort of dynamically CD24 expression change. Most thing from this environment, microenvironment, or the drug treatment. But I believe more this is heterogeneity from the sample, even from the same sample. We can see different CD24 expression even in the same genetic background.
That's so interesting. So in your initial study, you said you took the 60 samples and 28 were newly diagnosed and 32 were relapsed patients. And you were just looking for the levels of that CD24 expression to see. And that's so interesting that you just said that the application of drugs, some drugs can actually increase the level of CD24. That's really interesting.
Right. So that's a two-way. One is probably directly upregulated CD24 expression. Another one is the CD24 negative cells are killed. Only CD24 positive cells can survive. That's relatively enrich the CD24 positive cells. So we believe the later one enrich the CD24 positive cells more likely than upregulation CD24 expression. But we need more study to confirm this observation.
Yeah, and so overall, I mean, you're looking at these samples to say, and your hypothesis that just higher levels of CD24 expression means tougher or more, less, or more likelihood to relapse, I guess. Is that correct?
Right. So yes, the short answer is yes. We think the high percentage of CD24 positive myeloma cells are relatively insensitive or resistant to current treatment, so therapy, or even transplant. And those cells can survive and also can initiate a disease relapse. That's our bigger hypothesis, yes.
Right, and that's what's causing the relapse, because these cells are still sticking around, and they're still tumor initiating, and they're still replicating, and they're not being killed by the drugs that you're getting, right?
That's right.
Correct. So to give you one example, Jenny, like say you have a patient gets treatment with standard of care, which is Velcade, Revlimid, dexamethasone. Say they get treatment after 4 cycles, you see that they have not achieved adequate response. In that case, it would be worth looking at why they are not achieving response, because typically what happens is that patients they receive second line of treatment, which is usually Kyprolis Revdex, or say Kyprolis pomalidomide dexamethasone, or they go for transplant, or they go for daratumumab combinations. In spite of all these combinations, certain percentage of patients don't achieve MRD negativity, and they keep relapsing. In those patients, we believe that there will be increasing proportion of CD24 cells within the plasma cell, within the monoclonal plasma cells, which is what is keeping the persistent disease, which is what is keeping, which is what is leading to the relapse.
Right.
Yeah.
So what you mentioned is the first part of your study, and one of the goals of your project is to understand how much CD24 does a patient have. Are you separating that out between, again, like between newly diagnosed and relapsed patients? Or are you just going to look at relapsed patients, or who are you going to be testing?
We do. So in this proposal, the crowd, we proposed to test 60 newly diagnosed patients, but we'll also do the follow-up, collect the sample at a different stage, like we collect the sample at diagnosis, in remission after treatment, and also sample at relapse. So we can follow up this serial samples, see those changes, can support our hypothesis, or see if this really can be used as a marker to predict the patient response, or early relapse, or resistant, etc. That's our goal. Yeah.
And you're going to be using samples from existing University of Iowa patients, correct?
In this moment, yes. The reason is we have an intellectual property in process. Once we get this approved, I think we can that will be opened soon. Another thing is we have a manuscript is under in reversion in a relatively good journal is journal NCI. So we have to once this is published, then everything is for public.
Yeah. So right now we'll be doing it at University of Iowa, this testing, and then as Dr. Zhan mentioned, there is some intellectual property thing going on with it. We are hoping that it will be all cleared, and then the revision of our submitted manuscript is getting ready. So we are hoping that in next few months it will be ready for everybody.
Yeah, great. And then the second part of your study is to say, gosh, if we have a lot of CD24 in a particular patient, then what drug targets could go after that target?
Right.
As far as like
absolutely.
A monoclonal antibody, right?
Right. So that's exactly what the plan is. So like say we see a patient who is a high risk, as you mentioned, say biallelic 17p deletion, and then patient has not achieved MRD negativity, and then we see that they have increasing portion of CD24 in the monoclonal plasma cell. Then rather than just blindly going for salvage transplant or say some kind of newer modality, the best thing will be to target specifically the surface CD24 with bispecific or with using some other technique, and Dr. Zhan is working on that as well.
That's right.
Yeah, I think this idea of identifying the cause so you can impact the effect is really needed in myeloma, because right now when people relapse, well, they look at the genetics for sure, and that does have an impact, but there are other things that are potentially part of that whole process. So the more you can personalize it and say, oh, we do see higher levels of this CD24, and then, let's go after it with this particular drug, that would be fantastic.
Right. That's great. Yeah. So now actually we collaborate with professor at Washington U, Dr. John DiPerso, for developing CAR-T CD24, and also try to develop bispecific antibody that should have both CD1, CD8, Syndicate-1, and CD24 double positive antibody. So we are doing this work, but we need some time to work this out.
Right, that's what one of my questions was going to be. Are there any CD24 drugs that already exist or monoclonal antibodies that exist? But it sounds like you're going after a partnership to develop that and then test it.
Right. So last year there one paper published from a Chinese group. They published in the Oncotarget journal, named "Engineering a High Affinity Humanized Anti-CD24 Antibody to Target Hepatocellular Carcinoma, Liver Cancer." So but this is in preclinical mouse model, not in human sample yet. So currently, I don't think any relative mono CD24 antibody in cancer patient yet. However, because CD24 is a ligand for the I think it's E-selection. It's P-selectin, yes. And the anti-P-selectin is in treating sickle cell anemia. So I'm thinking the other antibody can block this binding between this ligand with this receptor, such as that drug like anti-P-selectin antibody maybe can be considered for using in treating cancer. That's one we needed to do, but we didn't start this experiment yet.
Well, that's really neat that you're looking at existing things that are being used in other diseases or other cancers and then being able to bring them to myeloma. That's more efficient than trying to create your own. But creating so at WashU, they're both creating a CAR T cell going after CD24 and a bispecific antibody?
Yes,
is that what I heard you say?
So we had a discussion. Yes, we had a discussion about 3 months ago. So they try to develop both ways.
Interesting. That is so fascinating. Right. Are they seeing other uses for that CD24 or just in myeloma?
In this one, certainly we consider more is myeloma because we don't know CD24 expression in other cancer. Of course, like breast cancer, people say the CD24 is negative in breast cancer stem cells. However, it's positive in liver cancer as a marker for liver cancer stem cells. So, and Dr. DiPeso, he is also is hematologist. So in this one, we think more is in myeloma, but that depends on future. If we test the CD24, could be a marker for other cancer or other disease. That we need to think more in the future.
Mm-hmm. So when you think about this, so if the CD24, let's say you develop this CAR T cell or this bispecific antibody or something like that can go after that target, what could you envision it? How could you envision it being used in the clinic? Like, would you give it up front as part of your induction therapy so that the cancer cells, like you're hitting certain cancer cells when you give a transplant or when you give RVD or something like that. You know you're hitting a lot, but maybe not all of it. So do you just add it at the front end, or do you get through that part until when the tumor burden is lower, and then you just say, okay, now we're going to give you a CD24 thing to kill off, these stem cells that keep proliferating?
So that's a very good question, Jenny, and this is the evolving strategy. So this is a very good question you asked, and it makes sense to use it upfront after the induction chemotherapy when there is minimum residual disease left, and target it with CD24 specific antibody, either a bispecific or a CAR T, so that we get them into we try to wipe out these tumor initiating cells upfront. So that's the strategy we are thinking about.
Yeah, it's a long time. Dr. Checker always educated me. Has said currently a lot of drug can induce patient in remission, but the problem is really patient relapse after a while in remission. So our goal is really develop a treatment to prevent the disease relapse. So directly answering your question is we think this treatment should be when the tumor burden is low after kill the most bulk tumor cells. Then it's a good time to add this specific treatment to eliminate sort of drug-resistant myeloma stem cells.
Well, that makes sense. You kill off most of everything, and then when the tumor burden's low, you go after that. I have a question going back to what you were saying before about because this is a flow cytometry test, and forgive me because I can't remember. That's just a blood test, right?
No, that's a bone marrow test.
It's a bone marrow test. Okay.
It's done on the bone marrow.
Okay. And does every flow cytometry test currently look at those CD24 levels, or is that something that you kind of have to specially request, or the lab has to really run?
So first of all, so when we do the flow cytometry, there are typical markers looked at. CD24 is not one of them. The individual lab has to standardize the test and then validate it and then run it. So even if, like say someone is in, say in Kansas, University of Kansas, and they request their hematopathologist that, can you please check CD24 fraction on myeloma cells? They might not be able to do it immediately because they need validation, making sure that the test is replicable and it's working well before they can start giving the results out. So these are the technical issues with the test. In our institute, we have overcome those issues right now, so we are able to do it routinely on the patients here at University of Iowa.
Well, that makes sense, and I think this could influence testing and treatment in the future if you find that this is true and that you have a target that can go after it. I think people would start changing their test to be able to predict for that, because then you have more personalized medicine.
Right. So this is basically a really personalized medicine where we are going after one particular marker to see how the things will span out in the future. So we are very excited about it.
Yeah, I think it's wonderful, and I know there are other doctors that are trying to go after the stem cells, and even in the CAR T environment, when most patients don't have a lot of CD19 on their myeloma cells, and so that one patient that had such a dramatic response with one of the first CAR T treatments at UPenn, they were really surprised that she had that response. But I remember them saying, oh, well, these might be some of these early precursor cells. So trying to find out why people are relapsing and going after it is truly amazing, because a lot of patients have really good responses with the treatments. It's just, it doesn't, over time they relapse. So if you could knock everything down and get rid of it all, that's really amazing.
Right. Yeah, so CD19 and CD24 both expressed in different stages of B cells, the B cell marker. However, they are not simultaneously expressed in the same stage. Sometimes they express CD19, sometimes CD24, or both. So currently we test 8 patients. When I worked with a colleague at the University of Utah, he found that 3 of 8 patients have both CD19 and CD24 expression.
Oh, interesting.
Only there are another 5 only express CD24, no CD19 expression. But the number is relatively small. We need to do more in the future.
Yeah, that's so amazing, because you could get to a point where you just do it individually for each patient and say, well, you're going to need this drug combination, you're going to need this drug combination. And that's, I think, so promising in myeloma and so exciting in myeloma that you're getting to this point. It's really amazing. So how long do you predict the study will take, and how long will you need to watch the patients? Just curious.
Right. So we started this clinic sample for doing flow cytometry almost 1 year, around 1 year, right?
Correct.
Yes.
Correct. Yeah.
So far we don't have clinical outcome yet because 1 year is too short to say good prognosis or bad prognosis. So our goal, once we first we have two goals. One is the prognostic marker, or drug will respond or not to using this CD24. Another one is if this correct, then we need to develop a treatment. So I think in the next probably 3 to 5 years, at least the one group in my lab will focus on this project. That's I think we need this time. Yeah.
Well, that's great. And then we talked about earlier in the show about how these projects will integrate with HealthTree. How do you envision this integrating with HealthTree?
So in my clinic, can you hear me? Sorry,
Yep.
I thought you were not able to hear me. Anyway, so in my clinic, we have started asking patients to register themselves on HealthTree, and they will be putting their own information. And then, as a part of HealthTree initiative, we have discussed about including this particular marker on their flow cytometry testing list, or in one of the sections, like say some kind of bar can be created, and then patients can ask, what is my CD24 number? And doctors can tell them that this is the number, and they can just themselves go ahead and put it there. And that way, there is a running data available. So there is already always updated data available for these patients. So that's the plan to integrate it with the HealthTree. So this will help in numerous ways. So the first way it will be, say patients can, say there are 500 patients with CD24 number newly diagnosed. What's happening with their treatment? How many are responding? What's happening to the CD24 number after their response? All this can be looked at with a more detail and with a broader view. So that's what we think we should be doing with the HealthTree initiative and to integrate it with the HealthTree.
And with HealthTree, you'll also be able to say, so we'll include CD24, and then you can also add a percentage. So this happens too on like genetic deletions and things like that. Not every patient has the same percentage. Like some patients might have 5% of CD24, and some patients might have 80% of CD24. And that percentage is kind of important, right?
Correct. That percentage is important as well. So HealthTree will be enormously beneficial for seeing the data. For example, we have 500 patients, we have their CD24 number. Like say out of that, 50 patients don't respond to the treatment. So what was their cytogenetic fish risk, and how did that correlate? Was there any correlation, or there wasn't any correlation? All those questions can be answered with the HealthTree data.
Yeah, I think it's really important. Well, we encourage patients who are listening to join HealthTree because this is a way that we can help the research community get to these types of conclusions faster. Now, I want to leave just a few minutes for maybe one or so questions because we're running short on time. But if you have a question for Dr. Zhan and Dr. Jethava, you can call 347-637-2631 and press 1 on your keypad. And our first question, 558-8163. Go ahead with your question.
Hi, thank you for having me on. Doctors, you mentioned that there's a debate about the existence of myeloma stem cells. I was just wondering why there's a debate at all, and why can't we just easily see whether they exist or not?
Sorry, I didn't understand your question. Can you repeat that, please?
Sorry, I was using my headphones. Can you hear me better?
Yeah, this is much better, yeah.
Okay, sorry about that. I was wondering why there's a controversy about the existence of myeloma stem cells.
So, first of all, it's very difficult to diagnose. So what is basically a feature of stemness? So the feature of stemness is that these are the cells which persist for long and they are responsible for relapse. So to characterize these cells, we need to have patients who have lived more than 10 years or 15 years or those who have had relapses. So we need to find this population in this particular cell population in group of patients who have been in remission for long to see what is the characteristic of these cells. Now, it's very difficult to find these kind of patients who have lived for so long. Plus, as Dr. Zhan mentioned initially, gene expression profiling and all these modern techniques were not available till 20 years ago, and now they are more and more available. So there are technical issues and there are logistical issues which has led to difficulty of characterizing these cells.
Yeah, simply say is, when we got the cell line in culture in vitro, 99% of cells are CD24 negative. However, these cells can divide and propagate proliferation. So people think every cell could be sort of stem cell feature. They can divide. That's one concept. And currently have two concepts for cancer stem cells. One is believe every cell could be cancer stem cell. Another one is like we propose that only a small population of tumor cells have stem cell features. So that's the reason some people believe the first concept, some people believe the second one. So from currently our data, when we collect CD24 positive cells from myeloma cell line, we can only use 10 cells generate tumor in mice. However, when we use CD24 negative cells, even 10,000 cells, no tumor formation. So that's big evidence support our concept, the later one, only a small population have stem cell features. I hope this can help you to understand a bit.
So what Dr.
Yeah, a little better. Thank you.
So what Dr. Zhan said, I'll probably repeat it. So, there are two concepts, two philosophies. One is that small population of stem cells lead to the cancer relapse and progression. Second philosophy is that every cancer cell is a stem cell. Now, we believe in the first philosophy that small percentage of cells lead to cancer generation, and that is based on our laboratory experiment. What Dr. Zhan's lab has shown that if you take CD24 positive cell line from myeloma patient cell line, only 10 cells are enough to produce myeloma. That's it. Now, if you take CD24 negative cells, and even if you inject, say, 10,000 cells in mice, it will not lead to the myeloma formation. So this is the reason why we believe that if even such a tiny number like 10 cell can lead to myeloma formation, we believe that this might be the marker. I mean, that's just now our hypothesis.
That's amazing. Okay, thank you for your answers and your question. Okay, one more question, 949-5572. Go ahead with your question.
Hi, Dr. Zhan. You mentioned that you had been tracking some patients at the University of Utah with the CD24 present, and I wasn't totally caught up in what you're saying. But my question is, have you been able to track the outcomes of those patients and how they responded to treatment?
Thank you for this question. Actually, short answer is no, because this is not done by myself. Is a collaborator, is a pathologist, Dr. Salama, who now he moved to Mayo Clinic, Minnesota. And also the patient information, we don't know. This is a de-identified information, so we don't know that part. Also, only 8 samples we did this analysis. We really didn't think we should follow up patient outcome because the small number size in this moment. But the future, yes, we should do that.
Right. So at University of Iowa now we have identified the patients who have MRD negativity and who have CD24 fraction positive in them, and we are following them. We have I cannot disclose everything right now, but we have sizable number of patients we are following closely, and we are monitoring their CD24 on bone marrow. And it's interesting because some of them, these patients have no disease biochemically. So biochemically, there is no M protein. They have normal kappa lambda ratio. But then when you do the bone marrow, and we have a very sophisticated MRD detection technique, which is by the Euroflow, the next generation flow, we do it here. And by next generation flow, we see that if they are MRD positive, and they have CD24 fraction, then we are closely following them to see what's going to happen. So it's evolving right now.
So this is exciting yet maddening, because most of us myeloma caregivers and patients do not care about the being hidden in a de-identified database. In fact, it's not really helping us. That information for those 8 de-identified patients is going to be forever lost somewhere. And you have in there some potential clues for a hypothesis that strengthens what you guys are doing. So I think this is really interesting in terms of how this does intersect with HealthTree, and how this could really accelerate this type of research and protect the patient's information, but yet not lose it in obscure IRB study.
Well, we totally appreciate your point. And I guess the issue is that when do we want to inform the community, right? I mean, we don't want to be going out and telling the information which we ourselves are not sure. And I think when Dr. Zhan and this research was done in Utah, at that time the patient number was so small that it was probably not the best thing to do anything with that. But I think in last 7-8 years, the research has become more mature and more information is available. So now I think in going forward, it will be very important to track this information.
Right. That's certain, yes.
Mm-hmm. Very good. Well, thank you.
Thank you.
Yeah, thank you for your question. Well, Dr. Zhan and Dr. Jethava, we're so appreciative for you, for the research that you're doing, and for joining us today to help explain it. And we're very, very excited about what you're doing. And it's clear because we selected you to receive a grant and to go after this for us. I think this has been a mystery for a long time, and I'm grateful that you have such deep experience working on it, and we're just so thankful that you're looking at this and studying it on our behalf.
Thank you all very much. Thanks, Jenny. You guys have a good day.
Thank you very much for your Jenny, and we appreciate your help. Thank you.
Well, thank you so much, and thank you so much for our listeners for listening to Myeloma Crowd Radio. And we invite you next time to learn more about the latest in myeloma research and what it means for you.
Step into the world of power, loyalty, and luck.
I'm gonna make him an offer he can't refuse.
With family, cannolis, and spins mean everything.
Now, you wanna get mixed up in the family business.
Introducing The Godfather at ChumbaCasino.com. Test your luck in the shadowy world of The Godfather slot.
Someday, I will call upon you to do a service for me.
Play The Godfather now at ChumbaCasino.com.
Welcome to the family.
VGW Group. No purchase necessary. Void where prohibited by law. See terms and conditions. 18+.